Okay, so it's my pleasure and my honor to talk just after Carrie and Italo. Thank you for the introduction, because you will make my life easier now to introduce the subject. And I would like to share with you some data that we have generated on lymphangiogenesis, and a part of this work has been done in collaboration with Carrie and Italo. So in the laboratory, we are mainly interested in tumor microenvironment, both in the primary tumor and in the secondary site, in the lymph node or in lung, for example. And we are mainly focusing our interest on angiogenesis, extracellular matrix remodeling, and more recently, since about a decade, we are working on lymphangiogenesis. As already mentioned, lymphangiogenesis, which occurs in the primary tumor, helps the tumor cells to disseminate to the lymph node. And you can see here a labeling of lymphatic vessels using protoplaning antibody D240, and showing that these vessels are colonized, invaded by tumor cells, which will migrate to the lymph node. And then further on, these cells can disseminate to other organs, such as the lung, for example. So a remodeling of the lymphatic vasculatures occurs in the primary tumors. Enlargement of collecting lymphatics also are observed, and I will present you some data on the remodeling of the lymphatic vasculature in the lymph node. And it is also shown now more and more than in distant organs such as lung, for example. Lymphangiogenesis can also occur and could participate also in the progression of the metastasis. So we were interested in studying the remodeling of the lymphatic vasculature in benign tissue, malignant tissue, and lymph node. This work has been done mainly in collaboration with Professor Frédéric Kridelka, who is a clinician, and who is mainly working on cervical cancer. So what we did is first to analyze the lymphatic vasculature in a benign tissue in the cervix. And as you all know, the cervix is composed of different epithelia, the exocervix, the endocervix, and between these two epithelia, there is a special zone which is called the transformation zone. This is an area where the HPV virus is infecting and transforming the epithelium. So this is the area where the tumor is developing. So when we look for the lymphatic staining here in those areas, and this was done by a quantification and a computerized analysis, we were surprised to see that we observed a huge amount of lymphatic vessels in this transformation zone. And upon quantification, you can see here that the density of the lymphatic vessels was much more important in this transformation zone as compared to the exocervix or endocervix, suggesting that the lymphatic vessels there form a specific environment, what we call a lymphangiogenic niche, and the role of this area and on this niche is really not known, but we know, for example, that in this region there are less dendritic cells which are arriving to the benign tissue and then to the tumor. And then when we looked further during the progression of the tumor, then we observed an enhancement of the lymphangiogenesis, and always we observed a close contact between, or a proximity between the tumor cells and the lymphatic vessels. But there are plenty of data showing that the tumor progression is associated with increased lymphatic vasculature density, and this is correlated to the lymph node status. So I would like mainly to emphasize here the studies that we have done on lymph node themselves to see whether the lymphatic vasculature is remodeled during the progression of the disease before or after the colonization by tumor cells. So in order to do this in collaboration with Frédéric Kridelka, we have generated a collection of samples containing different types of lymph node, either sentinel lymph nodes which are negative, in which we cannot find any tumor cells, metastatic lymph nodes, or distant lymph nodes which are negative and not colonized. by tumor cells. So what happens in terms of lymphangiogenesis? So you can see here the staining that we have used using protoplanin antibody. This is the binarization of the images, and we can see that in negative lymph node, in distant negative lymph nodes, the lymphatic vessels are, of course, mainly observed in the subcapsular area. And interestingly, in sentinel lymph nodes, which are negative and not colonized by any tumor cells, we can observe a higher density of lymphatic vessels, which are migrating deeper inside into the medullary area of the lymph node. And this lymphangiogenic response supports the concept of the pre-metastatic niche, suggesting that there is modification in the lymph node before the colonization by any tumor cells. And then when the metastatic cells are colonizing the lymph node, we observed often a close proximity between the tumor cells and the lymph nodes, suggesting that maybe the lymphatic endothelial cells are forming like a shield around the metastatic cells, protecting maybe themselves from the aggression by the lymphatic, from the lymphocytes. Importantly, we also showed that these lymphatic vessels came from a proliferation of the lymphatic endothelial cells, here as seen by Ki-67 staining. Surprisingly, when we analyze more carefully these lymphatic, these lymph nodes in a pre-metastatic stage, and when we characterize the cellular and humoral immune microenvironment, we observed a huge amount of CD24 lymphocyte B cells and PD1 also positivity in the germinal center. So we can observe the correlation, and this was quite surprising, a correlation between the lymphatic vessel density and this density of CD20 lymphocyte B and PD1 staining as well. We still not know what is the reason of this correlation, but it means that before any colonization by tumor cells, the lymphatic system and the whole lymph node is completely remodeled, preparing probably the cells and developing a permissive environment for the tumor cells, which probably will be protected from the attack by lymphocytes. So this is something that we are further studying. So this data show that the remodeling of lymphatic system occurs, of course, in the primary tumors, and even we may have specific niche in benign tissue, and occurs also in the lymph node. Then we started to develop a new system in order to reproduce in clinical models what is observed in the clinic. And for this, Maureen van der Velde and Melissa Garcia Caballero has developed what we call the air sponge assay. In this system, we are using a small fragment of a gelatin sponge, which is soaked with tumor cells or growth factors, and which is then implanted in the air of the mice. Why in the air of the mice? Because this is an area which is rich of lymphatic vessels, and the purpose was then to see the modifications of the sentinel lymph node, the lymph node draining this air system. So we are using here Lewis-Lung carcinoma cells, which are luciferase positive, enable us to track the cells and to follow them. You can see that after 4 weeks, this led to the generation of tumors. We can also use melanoma cells, for example. In blue, you can observe the formation of lymphatic vessels, which are invaded by tumor cells. And interestingly, 2 weeks after the injection of the sponge, we cannot see any presence of tumor cells in the lymph, but we observed after 4 weeks the colonization of luciferase positive cells in the lymph node, and this was further demonstrated by histological analysis. So after 2 weeks, we have a so-called pre-metastatic status, and after 4 weeks, we have a metastatic stage. And when we looked for the lymphatic vasculature in this lymph node, we reproduced exactly what was observed in the clinic. We observed huge amount of lymphangiogenesis in the sentinel lymph node in mice bearing tumor, while in the control animals we only see a small vascularization, lymphatic vascularization. So the density is much more important when the mice bear a tumor. So this reproduces what was observed in clinic, and later on at metastatic stage we can observe the colonization by tumor cells here, which is associated with further increase of lymphangiogenesis. And you will see that these models that have been recently published will be studied later on, will be used later on to go deeper into the metastatic understanding on lymphangiogenesis. So the remodeling occurs also in the lymph node, and this suggests that, of course, as already mentioned by Professor Alitano, lymphangiogenesis and the remodeling of lymphatic system is an important new therapeutic target. And as already mentioned, the main driver of lymphangiogenesis is VEGF-C. And I just remind you that VEGF-C interacts with homodimers of VEGFR-3, but also with heterodimers of R2 and R3. And how the homodimerization of R3 or heterodimerization R2 is balanced and controlled is poorly understood until now. Previously in the laboratory we have shown that the remodeling of type 1 collagen is extremely important during lymphangiogenesis. For angiogenesis, it's mainly the remodeling of the basement membrane, which is a key event. Here, regarding to lymphangiogenesis, it is the remodeling of the interstitial collagen, which is the rate-limiting event. We demonstrated this in different in vitro and in vivo system. And for example, here in a spheroid migration assay in which we are using lymphatic endothelial cells in vitro, we showed that these cells were able to degrade the collagen fibers as seen here, and this was really important in order to allow the cells to migrate. If we block the MMP2, an interstitial collagenase, then we completely block the migration of the cells. So this had been previously reported. And this prompted us to study the importance of under 180 receptor, also called urokinase receptor-associated protein, or UPAHAP. UPAHAP is a member of the mannose receptor, so it's an endocytic receptor, and it has been shown that this receptor is able to internalize fragments of collagens, which are further degraded into the lysosome intracellularly. So we were thinking that, okay, because the remodeling of type 1 collagen is important in lymphangiogenesis, maybe this receptor could be a contributor, a determinant in this process. And we knew also that this receptor was expressed by migrating cells such as cancer cells, macrophages, and fibroblasts, but its role in endothelial cells, either blood endothelial cells or lymphatic endothelial cells, was not known at all. So we hypothesized that, okay, this receptor could play a role by remodeling type 1 collagen. This was a nice hypothesis, but this was a too simplistic hypothesis, because now I will demonstrate that UPAHAP is indeed involved in lymphangiogenesis, but not through this mechanism. And so we have identified a new mechanism of action of this receptor. So science is really exciting because you can then postulate something and then demonstrate that you are wrong. So in order to demonstrate that UPAHAP is involved in lymphangiogenesis, we have applied different pathological models of lymphangiogenesis in UPAHAP knockout mice, which have been generated by Niels Behrens in Copenhagen. And in this system, we are inducing a corneal lymphangiogenesis by thermal cauterization of the cornea. And after 3 days, you can see that vessels are sprouting from the limb here. And in the knockout mice, we observe more sprouting events and more branching. And this was further supported after 5 days, when you can see here that in the knockout mice, we have much more vessels. It's not the length of the vessels which were more important, but mainly the branching events and the complexity of the vasculature. Importantly, angiogenesis, which was concomitant to lymphangiogenesis in this model, was not affected by UPAHAP, suggesting that this receptor plays a role in lymphangiogenesis but not angiogenesis. Although most of the people consider that what you found in angiogenesis can be transposed to lymphangiogenesis, but this is not the case here. And then we extended our study by again using the air sponge assay, in which we were embedding a PIMT mammary tumor cells, and then the collagen sponge were transplanted into the air. And this can lead to the formation of lymphatic vessels here. Do you have your pointer, please? Because so we observed a higher density of lymphatic. It's not working well. So we can observe the higher density of the lymphatic vessel here. Again, also much more branch, as I've seen by a 3D reconstruction of the vasculature. Thanks a lot. Be careful. And again, we did not observe any modification in the angiogenesis, supporting again that the UPAHAP plays a role in lymphangiogenesis, but not angiogenesis. As already previously mentioned, VEGF-C is the main driver interacting with VEGF-R3 homodimers or R2-R3 homodimers, heterodimers. And here we have compared the effect of different growth factors, VEGF-A, angiogenic factors, working mainly through its interaction with VEGF-R2. And we also use a mutated form of VEGF-C, which has been described by Cariali-Tallo, which only binds VEGF-R3. So please keep in mind this, because we will have to distinguish between the capacity to interact with homodimers or homodimers and heterodimers. And so again, we went back to the air sponge assay, in which we here soaked the sponge with different growth factors, and we observed only a difference between the wild type and the knockout mice in sponges soaked with VEGF-C, but not VEGF-A, or not the mutated form of VEGF-C, suggesting that the role of UPAHAP involves the contribution of VEGF-R2 and R3 heterodimers. Then we went to in vitro studies using lymphatic endothelial cells, which were silenced for UPAHAP expression by using scRNA, as you can see here. And we analyzed the migration of cells in a Boyden chamber assay towards VEGF-C, VEGF-A, or the mutated form of VEGF-C. And again, we observed only a reduction of migration towards a gradient of VEGF-C, but not for the other growth factors. And this was further validated in another model of directional migration, when you can see here that upon silencing of UPAHAP, the cells here in a gradient of VEGF-C, they are just turning around, and they are unable to migrate in direction to the VEGF-C gradient. So UPAHAP silencing disrupts the guidance of the cells specifically in a VEGF-C gradient, supporting again that it is only efficient in lymphangiogenesis and not in other systems. What about the mechanism of action of UPAHAP? We first looked whether UPAHAP can interact directly with VEGFR-2 and or R3. So we applied different techniques, co-immunoprecipitation assay or the proximity ligation assay, and we observed that UPAHAP can indeed directly interact with VEGFR-2. This is stimulated by VEGF-C, and it can also interact with VEGFR-3. But the more important finding is this one. We observed that upon silencing of UPAHAP, we observed much more heterodimerization of R2, R3, while we have no modification of the level of homodimerization R3, R3. And to confirm this data, we have performed the opposite experiment in which we overexpressed UPAHAP, and then we observed a reduction of the heterodimerization. Then we looked for the intracellular signaling to see how UPAHAP can affect the signaling. And there we were quite surprised because we did not see any modification of the classical pathway activated by VEGF-C, like ERK or AKT, but we observed a reduction, a modification here, reduction of the phosphorylation of JUNC, and thanks to the collaboration with Karhialitalo, we were able to demonstrate that this JUNC phosphorylation rely on a specific tyrosine residue here on VEGFR-3. And then we, after a long story, we were able to show that upon silencing of UPAHAP, when we have more heterodimerization of R2 and R3, this led to the phosphorylation of SERC2. And SERC2 is an adaptor molecule. When it is phosphorylated, you have intramolecular binding, and this inactivates this molecule. And what is the function of this SERC adapter? It is to be recruited here by the VEGFR-3 receptor, leading to the formation of complex with JUNC, which is then phosphorylated, leading to phosphorylation of paxillin and the control of RAC1 activation. And this GTPase is known to modulate the branching of the vessels and the migration of the cells. While in the absence of UPAHAP, the over-formation of heterodimers, so lead to the inactivation of SERC, and then affect the intracellular pathway activated by the VEGFR-3, and this led to an over-activation of RAC1 and hyperbranch lymphatic vessels. So this clearly shows a cross-talk which was, to my knowledge, not known between the heterodimers and the homodimers here. So meaning that UPAHAP is a new regulator, unexpected regulator of lymphangiogenesis, specific for lymphangiogenesis and not angiogenesis. It is involved only in VEGF-C-mediated lymphangiogenesis. It's a new partner for R2 and R3, and it acts as a gatekeeper, avoiding an excess of R2, R3 heterodimerization. So I hope that I have convinced you, and if I did not, I'm sure that Karhialitalo did it, that lymphangiogenesis is important new target which can play a role in different systems. And I would like to thank the people in the lab working on lymphangiogenesis, mainly the first authors of the papers that I presented, Tania Duré, Florent Morfoise, Cédric Bazin, Charlotte Erpicom. And I really specifically now thanks Tania Duré and Florent Morfoise, who are fighting with the reviewer to try to get the publication now in Nature Communication, and they are doing a fantastic job. I would like also to thank all the people in the lab, Nils Beren for providing us with the UPAHAP knockout mouse, and of course Karhialitalo, because we started working together more than 10 years ago. Our first shared publication was in Nature Methods, and hopefully we will have now this new paper together. Thank you very much, Karhi, and thanks to all of you.
For sharing enthusiasm for the difficulties in scientific research. So I'm afraid we are a little bit running out of time, so I would allow only one or two questions, burning questions, because it was really very clear and complementary to the first lecture. Yes.
Very nice, thank you. I was wondering whether the UPAR-associated receptor is involved in recycling, and what happens to the collagen binding activity when
So, yeah. So we have, first of all, we try to see whether we modify the internalization of the collagen or the degradation of the collagen, and we did not see any difference in the presence or absence of UPARAP. Also, it's important to mention that the effect that we observed is completely independent to the presence of the collagen. So we can do it in plastic or in classical condition. We do not need the collagen. So it means that, of course, UPARAP could play a role in vivo by remodeling the collagen, but in the system it's mainly the R2, R3 heterodimerization.
So thank you. Any other question? If not, then it's my duty to give you some reading about our academy.
Thank you very much.
So now it's my pleasure to